Okay, could somebody confirm you can hear me?
Yes.
Yeah.
Thank you. Before we begin, let me remind you, let me remind you that if you work together, you may work together, but if you do, everything you turn in should be in your own words. If you use the words of your study buddy, that will be plagiarism. And if you plagiarize something, you'll get a 0 on that assignment. Let me show you. It's actually in the syllabus, and I went over it the first day of class. You should be seeing my screen now. So today is actually February 1st, and we should be on chapter 4. Let me go. Student conduct. If I discover plagiarism, the use of another's work without citing credit, you will lose points and may score a 0. Students must, that's nothing important. Cheating is also prohibited during all quizzes and exams. But if you were to share questions and answers, that would be rather difficult. But if you were, you will get a 0 on that quiz or exam. Any questions about any of that? Plagiarism is important in college. Some ideas of how plagiarism, how important it is. There was once a sitting senator, I think he was the senator from Nebraska, and he was accused of plagiarizing something 20 years ago when he was writing up his PhD dissertation. And he had to resign his Senate seat. Another example, very famous example of plagiarism is Joe Biden, when he was first running for the presidency. I believe that year was 1988, but don't quote me on that. I'm not a history of politics. He was forced to stop running for the presidency because he plagiarized a British politician's speech. So plagiarism is important. Don't plagiarize. If ever you want to use your study buddy's words, you have to put it in quotes and say where you got it from. And then it's not plagiarism. Any questions about any of that?
Does this apply to our EC study group meetings?
The study group meetings, not if you're studying and you're quizzing each other. However, if you're working on an assignment together and then you both turn in the same wording, yes, that is plagiarism.
Got it.
Okay. All right, let's go with chapter 4. Oh, for those of you, let me call that back up. For those of you who are in the 7 p.m. lab, you'll note that there is no lab. Excuse me, there is no lab today. I will be logging in for 15 minutes just to answer questions, but after that point, I will log out. If you don't have questions, you do not need to show up. And a lot of people showed up for the 3 p.m. lab. And I had to tell them there's no lab scheduled for today. So I'm just here for questions. If you don't have any questions, you can log off. The same with the 7 p.m. lab. Just a minute, I'm having difficult time finding my find. Oh, there it is. So last time, did we end up on this slide talking about the gram-positive cell wall?
Yeah, I believe so.
Okay. Thank you. So the gram-positive cell wall has layers of peptidoglycan. It does have teichoic acid and lipoteichoic acid, which help the layers hold the layers of peptidoglycan together. The gram-negative cell wall, I assume we have not talked about that. Is that correct?
Yeah, you're correct.
Okay, great. Is a little different. First of all, it has a very thin layer of peptidoglycan, and that layer is in between two layers of lipid, meaning there's a cell membrane below the cell wall, and it's a lipid bilayer. Let me blow this up. It's a lipid bilayer containing mostly phospholipid. And then we have the cell wall, which does include this outer membrane of the cell wall, but that outer membrane of the cell wall is a lipid bilayer. In fact, this layer of the bilayer is essentially the same as the cell membrane. The main molecule is phospholipid. This lipid bilayer is a little different. Come on, mouse. The upper one of the outer membrane. It's probably the main molecule is phospholipid, but it does have another lipid in it, lipid A, and attached to lipid A is an O polysaccharide, so that we call this molecule with lipid A and the O polysaccharide a lipopolysaccharide. The O polysaccharide makes the hair, the big blue blob is the lipid A. Lipid A is important because it is an endotoxin. Toxins are something which is toxic to us. And so the outer membrane of the gram-negative bacteria cell wall contains an endotoxin. And that means if you ever have a gram-negative bacterial infection that has a cell wall, you will be exposed to endotoxin. Any question about any of that? Let me see if I can get that out of the way. The outer membrane has another important protein in it, and it's the porin protein. And there are other molecules in the outer membrane. There are peptides shown here. There are lipoproteins shown here. And a lipoprotein is a protein where the protein portion is generally in the peptidoglycan portion. And then the lipids are in the either the outer membrane of the cell wall or in the cell membrane itself. And there can be carbohydrates in it as well. Obviously, the O polysaccharide is one carbohydrate. The porin protein is important because it is a channel protein that allows molecules that the cell wants to enter the outer membrane, meaning most molecules cannot move through this lipid bilayer of the outer membrane. There are some exceptions, like a fat-soluble molecule or a lipid-soluble molecule, like cholesterol, can obviously move through it. And then carbon dioxide and oxygen can move through it. All small nonpolar molecules can diffuse through it. But other molecules like glucose, fructose, amino acids, all of those important molecules, they will not move through the lipid portion of the outer membrane, and they do not move through the cell membrane either. Now, the cell membrane, how it gets the molecules into it that it wants is it makes a transport protein to move one molecule across the cell membrane. And so there's a separate transport protein to move fructose, and it's a different transport protein than the transport protein to move glucose. And that's how molecules enter and leave the cell membrane, through a transport protein, unless they can diffuse through it. That's not the way things move through the outer membrane. They move through the outer membrane through this porin protein. And it is a protein that creates a pore, and this pore allows molecules to enter that the cell wants, and it allows molecules to leave that the cell wants to leave. It does not allow all molecules to enter. For example, penicillin G does not get through the porin protein, and it cannot get through the outer membrane lipid bilayer. Come on, mouse. There we go, the lipid bilayer. So if it can't get through the outer membrane, it does not get into the cell wall to damage the cell wall. And penicillin G does not work against gram-negative cells. Any question about any of that? So both amino acids, fructose, glucose, all of those molecules the cell wants come through the porin protein of the outer membrane. Just looking to see if I should say anything further. I'm not going to quiz you on this, but the periplasm is the part of the cell wall between the outer membrane and the cell membrane. And that includes the area of the cell wall where the peptidoglycan is. But remember, the cell wall is both the peptidoglycan and the outer membrane of the cell wall. So in the outer membrane, we have lipopolysaccharides. That includes lipid A. There are lipoproteins, and that's just a protein which runs through either the outer membrane or the cell membrane, as well as the peptidoglycan layer. And then phospholipids. The outer membrane of the cell wall can protect the gram-negative cell from complement proteins. This is a protein that we make and we have in our blood, and it does attack foreign microbes, but it cannot get through the outer membrane. So the complement proteins don't work well, if at all, on the gram-negative cells. And then certain antibiotics like penicillin G cannot get through the outer membrane. The outer membrane does have the old polysaccharide antigen, and that's part of the lipopolysaccharide. That antigen is actually what gives E. coli 0157H7 part of its pathogenicity. And that is, E. coli is a bug that doesn't cause us much harm, and we have lots of it in our gut. However, there are variants of E. coli which are human pathogens. And E. coli 0157H7 is a variant which is a human pathogen. The 0157 is an antigen, H7 is an antigen. 0157 is part of this O polysaccharide, which is right there, which is part of the lipopolysaccharide containing lipid A. And then the most important thing to remember with the gram-negative cell wall is it does contain lipid A, which is an endotoxin. All right, when we talk about the gram-negative cell compared to the gram-positive cell walls, they do differ. The gram-positive cell wall has a thick peptidoglycan structure. It's a more rigid cell, meaning that thick peptidoglycan makes the cell rigid. The gram-negative cell wall has a thinner peptidoglycan layer, and assuming the gram-negative cell has a cell wall, it does have peptidoglycan. You might have noticed the way I worded that, and that is there are some, just a few gram-negative bacteria that lack a cell wall. And these cells that lack a cell wall stain gram-negatively. And that's because the crystal violet iodine complex does not get caught in the thick peptidoglycan layer. So they stain gram-negatively. But most gram-negative cells do have a gram-negative cell wall. It is a thinner peptidoglycan layer. It is rigid, but not as rigid as the gram-positive cells, meaning the cell wall does protect the cell and does give the cell shape, but it's not as rigid as the gram-positive cell. Teichoic acid is found in the gram-positive cells. There is no teichoic acid in the gram-negative cells. There is no outer membrane in the gram-positive cell wall. There is an outer membrane with lipid A in the gram-negative cells, and lipid A is an endotoxin. Any questions about any of that? All right. So let's talk about the gram staining mechanism. Crystal violet iodine crystals form a complex that forms and will color the cell, staining the peptidoglycan. In the gram-positive cells, this crystal violet iodine crystal does not leave the thick peptidoglycan layer. In the gram-negative cells, the decolorizing agent washes the crystal violet iodine complex out of the thinner peptidoglycan layer, and the alcohol will dissolve the outer membrane. The outer membrane is mostly lipid, and so it'll get dissolved by the alcohol of the decolorizing agent. And then safranin will stain the cells pink. Why the gram-negative cell is staining pink, and the gram-positive is staining purple. So looking at the gram-positive and the gram-negative, you can ignore all the lower portion of this table. The gram-positive is going to retain the crystal violet iodine complex. The gram-negative will lose it. The gram-positive has a thick, multi-layered layer of peptidoglycan. The gram-negative has a thin layer. It's not single layered. This is a mistake of the table. It's at least 3 layers of peptidoglycan, but it is thin. It's between 3 and 30. The teichoic acid is present in gram-positive cells, absent in gram-negative. The periplasmic space is absent in gram-positive cells, present in gram-negative. The outer membrane is absent in gram-positive cells, present in gram-negative cells. The lipopolysaccharide content, there's none in the gram-positive, and it's at high levels in the gram-negative. Now, there are some cells which have an atypical cell wall. What we've been talking about are bacteria. So let's talk about the archaea first. It has an atypical cell wall compared to the bacteria, meaning the cell wall of archaea are based upon pseudomurine. It's another disaccharide. it is not based upon peptidoglycan. And some archaea lack a cell wall. Okay, which obviously some bacteria lack a cell wall as well. Anyways, the archaea differ, and that's not surprising because they're in a different domain from the bacteria. Inside the bacteria, there are two families which have atypical cell walls. There's the mycoplasmas, and this family of mycoplasma lacks cell walls. These cells tend to be pleomorphic in shape because they do not have a cell wall to give the cell shape. The mycoplasmas, interestingly, have a sterol, which is in their plasma membrane, and it's believed the sterol gives the plasma membrane strength and support, much like animal cells have cholesterol in their cell membrane, which also gives the cell strength and support. And it's thought that these sterols are just in the cell membrane to give it a little more strength because there's no cell wall to protect the cell membrane. All right, there's another family of bacteria which has an atypical cell wall, and that's the Mycobacterium. Don't get the Mycobacterium mixed up with the mycoplasmas. Mycoplasmas, no cell wall. The Mycobacteria does have a cell wall, and it is a gram-positive cell. However, it has a lot of macolic acid in the cell wall. That macolic acid makes the Mycobacterium an acid-fast staining cell, meaning you put an acid-fast stain in the cell, it'll stay there, it will not decolorize. And so we say that Mycobacterium are acid-fast positive cells. And then almost all other bacteria are acid-fast negative cells. We'll talk a little bit more about the Mycobacterium later. I don't think it's in this lesson. I think it's in a later lesson. And I think we've talked about it at one other place, either in the lab or the lecture. But we will talk a little bit more about it in the future. Maybe I should briefly show you. I don't have a picture here, so let me go to Google. Macolic. Show you a picture of the macolic acid in the cell wall. So here we have the cell wall, and you can see peptidoglycan right there. And it has a large amount of macolic acid right there. And that macolic acid will keep water-soluble molecules out of this cell. And it will also keep water that's inside the cell from leaving the cells, so that these mycobacterium are resistant to drying out, and they're resistant to many other chemicals that are water-soluble because they can't get inside the cell. Any question about any of that? Maybe that's a better picture here. Yeah, it's a better picture here. So peptidoglycan layer of the cell wall. Like I said, these are a gram-positive cell, but they're different. And that is they have this molecule here and then a very thick layer of macolic acid, which is a lipid, and it doesn't allow water-soluble molecules to move into the cell. Probably should have saved that down for the future, but Someplace I've got a picture of it, but it's not in this lesson. All right, you can damage prokaryote cell walls. We're not going to talk much about it, but you could dissolve the cell wall in a bacteria with the enzyme lysozyme. It digests the peptidoglycan. And you don't need to know what the cells are called when you remove the cell walls. You can also damage the cell wall with penicillin. It inhibits the peptide bridges forming in the peptidoglycan, creating holes in the cell wall, and the cytoplasm or the cell membrane can protrude out of it. And without the cell wall being intact, meaning if it has holes in it, it's not protecting the cell. Because of the difference between the cell walls of the gram-negative and the gram-positive cells, I need to mention the gram-negative bacteria are not as susceptible to penicillin G and similar antibiotics like penicillin V. I think that was the second penicillin member of the penicillin family discovered. And then some of the cephalosporins as well. However, that doesn't mean that gram-negative cells are not susceptible to antibiotics. There are other antibiotics that can penetrate the outer membrane of the gram-negative cell. For example, ampicillin, which is one of the early antibiotics discovered, like I think it was around the third member of the penicillin family. It does get through the outer membrane of the gram-negative cell. So ampicillin can work on both gram-negative and gram-positive bacteria. Most of the later members of the penicillin family can work on the gram-negative as well as the gram-positive. And there are other antibiotics like streptomycin. This is a lipid-soluble antibiotic. So it gets right through the gram-negative cell wall. Streptomycin is also an antibiotic that can get through the malic acid of the mycobacteria. And this is an antibiotic which is frequently used against a mycobacteria infection. All right, any other questions about the cell wall? If not, let's start talking about the cell membrane. The cell membrane is also called the plasma membrane, and those two words are used interchangeably. It is mostly a phospholipid bilayer shaped like this, where the heads face inside the cell. So the heads, which is containing the phosphate portion of the phospholipid, is facing mostly the water of the cytoplasm. The fatty acid tails, which are lipid soluble, are on the inside of the cell membrane. And then we have another layer on this way facing that way with the tails on the inside and then the heads facing outside. You have to remember that in a person or any living organism, that generally we're about 70% water. And so most of the material outside the cell is also water. And if you think about a red blood cell floating in the blood is mostly water. And so it has the phosphate heads facing the mostly outside water. You do have proteins in the cell membrane. And there's actually three types of proteins. There are peripheral proteins, integral proteins, and transmembrane proteins. The peripheral proteins are proteins which are mostly on one side of the membrane or the other. That's why it's called peripherals, peripheral to the cell membrane. the integral proteins and the transmembrane proteins both span the membrane. And an example of a integral protein shown here is a transport protein, which of course has to span the membrane because it allows a molecule to enter the cell by transporting it across the cell membrane. There are other molecules in the plasma membrane, like various carbohydrates. Most of these proteins are free to float about the cell membrane. So those of you who've taken anatomy and physiology know that when a ligand binds to a receptor, and the receptor is a protein, that the receptors move together in the cell before the receptor is brought inside to bring the signal into the cell. Okay, so my point is that the proteins can move about in the cell membrane. That means that the cell membrane is actually a fluid. And it's hard to think about it when we can look at my hand here, for example. It's held together by the cells, which are held together by cell membranes. And that cell membrane is actually a fluid. It is as fluid as olive oil. I assume olive oil at room temperature, because if you put olive oil in the refrigerators, it's a solid. But the membrane is a mosaic of phospholipids and proteins, and these proteins move about in the cell membrane to function, like the receptors protein that binds to a ligand. It will move together before it be brought into the cell. And so to function, that protein has to move about. Not all proteins are free to move about the cell membrane. If the protein is anchored to the intracellular matrix, that is a matrix of proteins running through the cytoplasm of the cell, and it gives the cell a backbone. If that protein, let's say this one here, is connected to the cytoskeleton, that will anchor this protein in place, and it is not free to move about the membrane. Another way a protein can be anchored in place if it is anchored to an extracellular matrix, mainly protein. So if this protein here, which I'm sorry, I should really take another protein, this protein here, if it's anchored to the extracellular matrix, this protein will not move about in the cell membrane. And you can think of a protein in the cell membrane of a cell in the bone being anchored to the extracellular matrix of the bone. And so that protein will not move about the cell membrane. But many proteins are free to move about the membrane. The phospholipids are also free to move laterally around the membrane, and they're also free to rotate, spin around right here. Okay, that is actually how water can move into the cell. Water does not move through the phospholipid bilayer, but water just comes up to the membrane. Let's make this pointer here. And then it waits for this fatty acid to move over to here. The water then sneaks in, and then it's in between the two phospholipids right here and here. It can't move through this layer of the phospholipid, but what the water does is it just waits inside this portion of the bilayer, and then it just waits for this phospholipid to move over here. and then the water moves inside the cell. And that is how water can move through the cell membrane. It's not a very efficient way of moving water across the cell membrane, because obviously it can move inside the cell or outside the cell that way. And for like roots, for example, in plants, they have aquaporins, which are transport molecules, transport proteins to bring water into the cell. And many cells have aquaporins, which are transport proteins to move water generally into the cell. Any question about any of that? The plasma membrane shows selective permeability. That means it allows the passage of some molecules, but not other molecules. No large molecules can move across the plasma membrane unless the molecule has a transport protein to transport across the cell membrane. Ions either do not move through the lipid bilayer, or if they do move, they move very slowly across it. And water moves slowly across it as well. So the smaller molecules can move through. I explained how water does it. Oxygen and carbon dioxide can diffuse through the lipid bilayer. If you think about oxygen, it's going to be pulling on its electrons equally. So it's a non-polar molecule, and it is small non-polar molecules, mainly oxygen and carbon dioxide, can move right through this lipid bilayer. Another molecule which can move through the lipid bilayer are a lipid. Lipids can move right through the lipid bilayer because obviously lipids dissolve in lipids. But if you think about it, there are not that many lipids in the blood. There are some, but there are not very many. The majority of the molecules in the blood are either water or a molecule dissolved in water. So lipid molecules can move through, a lipid soluble molecule can move through the lipid bilayer of the cell membrane as well. So all the molecules that the cell wants, like glucose and fructose, amino acids, nucleic acids, you have to have a transport protein to bring that molecule into the cell. The plasma membrane is also unique in bacteria in that it contains enzymes for making ATP. For photosynthetic bacteria, the photosynthetic pigments are found in foldings of the cell membrane. And here we're seeing a photosynthetic bacteria. Let me see if I can blow that up a little. And the cell membrane is not underneath the cell wall running like this. The cell membrane actually runs like this. So it's running like this, and then it's making a fold. Then running like that, then making another fold. Running like that, making another fold. Any questions about that? Oh, let me shrink that down. We call those things chromatophores or thylakoids. Those are the foldings of the cell membrane in a photosynthetic bacteria containing the photosynthetic pigments. You can damage the plasma membrane with either alcohol, quaternary ammonium compounds, or the polymyxin antibiotics, like polymyxin B, will put a hole in the cell membrane. That will cause leakage of the cytoplasm out of the cell, and that will kill the cell. Any question about any of that? I lost my, there we go. All right, let's talk about the movement of materials across the membrane. Materials can move across the membrane, and it is critical to cell that materials do move across the cell membrane. There are two ways to move material across the cell membrane. There's a passive process and there's an active process. A passive process is where the molecules move down its concentration gradient, meaning the molecules move from areas of high concentration to areas of low concentration. There are three ways that passive movement can occur: by simple diffusion, which a solid moves by simple diffusion, and by facilitated diffusion, once again, a solid is moving down its concentration gradient, and then by osmosis. Osmosis is where the solvent, mainly water, moves across the plasma membrane. And osmosis always has to involve movement of water across the membrane, either an artificial or a real membrane like the cell membrane. Something else I want to talk about osmosis. Osmosis is a solvent, not a solute. And water moves from areas of high concentration to where there's more water to where there's less water. Any question about that? All right. There are also the active process, and this is where molecules move from areas of low concentration to areas of high concentration, meaning the molecules are moving against their concentration gradient. How this happens is the molecule moves across the cell membrane using a transport protein that transports the molecule across the membrane, either into or out of the cell, and that requires the cell to spend energy. And for one molecule that moves across the cell membrane, the cell uses up one molecule of ATP. The ATP will be providing the energy to move that molecule against its concentration gradient. Any questions about any of that? All right, let's talk a little bit more about passive processes. This is when the molecules move down their concentration gradient from an area where they are highly concentrated to areas where they are less concentrated. Simple diffusion is movement of a solid from areas of high concentration to an area of low concentration, such as when we put a drop of dye in a beaker of water. The molecules of the dye will diffuse into the beaker of water, moving from areas of high concentration to areas of low concentration. And that process will continue until the dye reaches equilibrium, meaning there is no area that is high in concentration and no area that is low in concentration. An example of simple diffusion is the movement of oxygen and carbon dioxide across the cell membrane. We need that for life. We need oxygen to move into the cell, and it moves from areas of high concentration outside the cell into areas of low concentration inside the cell. And carbon dioxide moves in the reverse from areas of high concentration inside the cell to areas of low concentration outside the cell. Any question about any of that? All right, let's talk about facilitated diffusion. The facilitated diffusion is diffusion of a solute across a membrane, but this diffusion is facilitated by a protein. and we're going to call it a transporter protein. I tend to just call it a channel protein. And what it is this protein makes a channel in the membrane, allowing one molecule to move through the membrane. Fructose is a molecule which moves by facilitated diffusion into the cell. So if you eat a bunch of fruit, you'll have a bunch of fructose outside your gut cells, and that fructose molecule will move through a channel protein by facilitated diffusion from areas of high concentration to areas of low concentration. And so glucose isn't a good example, fructose is better. This means that fructose will move into the cell as long as there's less fructose inside the cell than outside the cell. Once the concentration of fructose outside the cell equals the concentration of fructose inside the cell, you will no longer digest fructose because fructose will stop moving into the cell. Any question about any of that? That means when you eat fruit, you will never digest all of the fructose, because once the fructose outside the cell equals the concentration inside the cell, fructose will stop moving into the cell, because that's the way facilitated diffusion works. That's different than glucose. If you were to eat glucose, which you can get from the digestion of table sugar, which makes fructose and glucose. If you were to eat sugar making glucose, which almost any sugar will break down into glucose, you have the enzymes in your gut to break down like maltose into 2 molecules of glucose. If you were to eat glucose, for example, all of it would be digested because initially glucose will move into the cell by facilitated diffusion using this protein. However, once the glucose concentration equals the concentration outside, so once the concentration inside the cell equals the concentration outside the cell, glucose can no longer move into the cell by facilitated diffusion. And then this transport protein changes from being a facilitated diffusion protein to an active transport protein. And then that protein will pump every glucose molecule into the cell until you have 0 glucose molecules outside the cell. And that's just the way glucose works. It initially works as a facilitated diffusion protein, but then it switches to active transport protein to pump in all of the glucose into the cell. So you will always digest all of the glucose that you eat. Any question about any of that? We'll talk more about active transport in just a little bit. So in both simple and facilitated diffusion, solutes are transported, and the solute molecule transports down a concentration gradient, meaning the molecules move from areas of high concentration to areas of low concentration. The main difference is that facilitated diffusion has a channel protein to move the molecule across the cell membrane. And then facilitated diffusion always involves a cell membrane. Simple diffusion may or may not involve the cell membrane. As this example is showing here, we have dye diffusing into the water. There's no membrane there. But I gave the example of oxygen diffusing into the cell, in which case there is a membrane that the oxygen diffuses through. Osmosis is the net movement of water down its concentration gradient across the selectively permeable membrane. Water is not a solute, it is a solvent. And although water moves from areas of high concentration to areas of low concentration, we tend to define the tonicity not by the concentration of water, but by the concentration of the solute. When we're talking about tonicity, a bacterial cell can experience three kinds of osmotic solutions: an isotonic solution, a hypotonic solution, and a hypertonic solution. You should note that each of these are relative terms, so it's correct to say that the solution outside the cell was hypertonic relative to the solution inside the cell. Isotonic is the easiest to explain, and that is where the concentration of the solutes outside the cell is equal to the concentration of solute inside the cell. If the concentration of solutes is equal, the concentration of water is equal, meaning the concentration of water outside the cell is going to be equal to the concentration of water inside the cell. So the cell is happy because there's no net movement of water. And note that we're talking about the tonicity by the concentration of the solute, but in this case, it is the solvent, which is the molecule which can move into or out of the cell. Everyone got that? All right. The cell is happy because there's no loss of water, there's no gaining of water. The cell can be in a hypotonic solution. This is where the concentration of the solute outside the cell is lower than the concentration of the solute inside the cell. Because of this, there is net movement of water into the cell. Where the solute concentration is low, that means the concentration of water is high. So there's more water outside the cell than inside the cell. And so water will move into the cell. Once again, we're defining the tonicity by the concentration of the solute, but in this case, it's the water that's going to move into the cell. As water moves into the cell, that will swell the cell so that the cell membrane will expand up until it reaches the cell wall. Once the cell membrane reaches the cell wall, the cell membrane cannot expand any further. If it cannot expand any further, there's no room for water to move into the cell. Even though water would like to move into the cell, it can't because there's no room for it to move into the cell because the cell membrane cannot expand any further. That will protect the cell because the cell membrane cannot expand any further than the cell wall. If the cell does not have a cell wall, like a human red blood cell put into a beaker of water, or a cell, bacteria cell, that has holes in the cell membrane because of peptidoglycan G creating the holes in the cell membrane, then water can continue to move into the cell. And the cell membrane will expand. In the case of the red blood cell, it will just keep on expanding and expanding until the cell membrane pops. And then that'll leak out the cytoplasm, killing the cell. In the case of bacteria having holes in their cell wall, water will move into the bacteria, and then the cell membrane will protrude out of the hole in the cell wall. More water will move into the cell, and that cell membrane will protrude further out of the hole. And it will protrude further until the cell membrane pops. And then that will leak the cytoplasm out of the cell, killing the cell. All right, any question about that? So when a cell is put into a hypotonic solution, if the cell does not have an intact cell wall, the cell will burst as the cell membrane will expand until it pops. And this we call osmotic lysis. This is actually how penicillin G kills a bacteria cell. It puts a hole in the cell membrane, and then the cell membrane protrudes out of the hole in the cell wall, and then more water will move into the cell, and the cell membrane will protrude further out of that hole until the cytoplasm pops. The cell membrane pops, I guess I should say, not the cytoplasm. Most bacteria live in a hypotonic solution, so they require their cell wall to protect them. Any question about any of that? All right, a bacteria can also be in a hypertonic solution. This is where the concentration of solute outside the cell is higher than the concentration of solute inside the cell. That will cause water to move from areas of high concentration inside the cell to areas of less concentration outside the cell. So water will move out of the cell. That will make the cell membrane shrink and pull away from the cell wall. This we call plasmolysis. And this generally leads to cell death, like E. coli in a hypertonic solution will not survive very long because water will leave the cell. An example would be E. coli that falls into a bottle of molasses. The water will leave the E. coli, entering the molasses, and that will kill the bacteria. That is why molasses can stay at room temperature for years. I don't know if you've ever had a bottle of molasses, but it keeps for a long time. My mother had a bottle of molasses that we figured was at least 20 years old, and it was still good. I made molasses candy out of it and gave it to my father, and he loved it and didn't get sick or die from it either. And finally, after we figured that the bottle of molasses was 30 years old or older, my mother decided to throw it away because neither she was using it or I was using it, and that molasses was really old. But as far as we knew, that molasses was still good. I think what she did was she bought the molasses to make pork and beans, which she stopped making obviously 30 years ago or longer. Plasmolysis generally leads to cell death. Any question about any of that? All right, the movement by passive processes is okay when the concentration of a substance required by the cell is higher outside the cell than inside the cell. But what about when the cell is in an environment with low nutrient concentrations? For example, a cell in the ocean. It's going to have much more nutrients inside the cell than the ocean water. Or a cell in the Sahara Desert. The cell is going to have more nutrients inside it than it's going to find in the desert sand. So in these cases, we need an active transport movement to move material against its concentration gradient. So cells must accumulate nutrients against their concentration gradient. This is going to require the cell to spend energy. The cell needs to perform work to move that molecule against its concentration gradient. How the cells do this is they use an active transport protein to pump the solute molecule against its concentration gradient. So the transport protein will be in the cell membrane, and generally it's molecule specific, meaning we have one transport molecule, one transport protein to move one molecule. And that would be something like the amino acid lysine. To move another amino acid, like the amino acid arginine, you need another transport protein to transport that one amino acid. To transport one molecule into the cell requires the cell to spend or use up one ATP molecule, adenosine triphosphate. It takes energy to move that molecule against its concentration gradient, and that energy comes from ATP. The cell splits ATP to get the energy it needs to move the molecule against its concentration gradient. Any questions about any of that? All right. In prokaryotes, we have a special form of active transport, which is called group translocation. It's a special form of active transport found only in prokaryotes, meaning only in bacteria and archaea. It does require a transport protein inside a membrane like the cell membrane. It does require energy, and it does not get this energy from ATP. It gets it from a high energy phosphate compound such as PEP. And then the molecule which is transported is chemically altered. So like glucose coming in by group translocation, the glucose molecule outside the cell will be pumped into the cell. PEP will be used to get the energy to move the glucose against its concentration gradient. And the phosphate group from PEP will be transferred from PEP to glucose. And so instead of getting glucose moved into the cell, it will be glucose 6-phosphate inside the cell. Let me see how I can talk about this. Blow this up. So let's say we have 10 molecules. Why is that not working? 10 molecules of glucose outside the cell. If all of them were to move into the cell by active transport, you need to pump, and let's say I have to say that we're just going to make it simple and say there's 10 molecules of glucose inside the cell. If all of the 10 were to move by transport, the first glucose, will be moving into the cell And it will only be 1 moving against 10. So how do you say that? Let's go against moving into the cell against 10 molecules. And then the second, moving into the cell let's go ahead and get that, although it's not. Oh, come on. It's going to be against 11. That's a stiffer concentration gradient than that one. And then the last one, it's going to be the 10th. And it's going to be moving something against, it's going to be moving against something like 20, which is a stiffer concentration gradient. Hopefully you guys see all that. So that was what would happen if we moved everything by active transport. But let's say we now move 5 into the cell by group translocation. And 5 into let's see where I got this. By active transport. If the fifth one that we're moving into the cell by active transport, so let's go to the fifth one. It's moving against well, it's going to be moving against 14 because we started with 10 inside the cell. Anyways, the movement of glucose into the cell will be 1 against 14, and that's less than 1 against 20. So it's thought that bacteria cells can split the movement by active transport and group translocation to make it easier to move the glucose into the cell. Hopefully you guys kind of understand this. I've tried to go through it a little bit, but the point is that it's easier moving a molecule into the cell because half are moved by group translocation and half are moved by active transport, as opposed to moving all of them by active transport. Any question about any of that? So it's thought that group translocation helps the cell accumulate substances because we're moving the molecule against its concentration gradient. And by altering the molecule in group translocation, you make 2 different types of molecules. There's both glucose inside the cell and now glucose 6-phosphate inside the cell. And I guess we could point that out here. And that is for the active, the group translocation, it's even better for this one. And that is the fifth one moving into the cell. It's only against 4. So instead of being against 20, the fifth one moving into the cell will only be moving against 4 because there are 4 glucose 6-phosphates inside the cell, and we're going to be adding a fifth now. Any question about any of that? Oh, excuse me. Oh, hopefully you didn't see any of that.
Yes.
Thank you. So group translocation is a special act of transport found only in prokaryote cell. Gonna sneeze here. Actually, that is normal that I usually sneeze twice, so it's not surprising. I somehow was able to hold that one back. Means I'll probably sneeze in the future. All right, let's talk about the cytoplasm. The cytoplasm is the substance inside the cell membrane. It's mostly water. About 80% of the cytoplasm is water. The other 20% are things dissolved in the water, and we call those solutes. That can be sugars, amino acids, proteins. DNA is actually dissolved in the cytoplasm. The nucleoid region is the region of the cytoplasm containing the DNA, and it is wherever the DNA is floating in the cytoplasm. There is no distinction or no boundary between the cytoplasm and where the DNA is floating. Ribosomes and inclusions are included in the cytoplasm, but these things are so big that they are not in solution. Let me see if I can blow this up. There's an inclusion. These little dots are ribosomes. And like I said, both of these are so big, they are not in solution. But they are found within the cytoplasm. The nucleoid region is simply wherever the DNA is floating in the cytoplasm. There's no nuclear membrane to separate the cytoplasm from the nucleoid region. Cells can also have a plasmid, a small molecule of DNA in the cytoplasm. And we'll talk more about that when we talk about genetics. Ribosomes are the site of protein synthesis. They are actually made up of 2 subunits. You don't need to know the sizes. Ribosomes are made up of both proteins, we call that ribosomal protein, and ribosomal RNA, or rRNA. And those are the 2 things that make up a ribosome. Prokaryote ribosomes are smaller than eukaryote ribosomes. So prokaryote ribosomes are 70S in size. Eukaryote ribosomes are 80S in size. The S just refers to where the ribosome sediments in an ultracentrifuge gradient. I'm sure that meant a lot to you, but that's what the S stands for. And all you need to know is it's just the size of the ribosome. Inclusions are reserve deposits that are brought into the cytoplasm, and the cell uses it when its resources are low. So something that the cell wants inside it, it will accumulate the material inside the cell until the point where there is so much material that the material precipitates out of solution. Once it precipitates out of solution, we then call it an inclusion. Any questions about that? So you see that inclusion, whatever the cell was bringing into the cell and storing, Initially, it would be in solution, but when it gets too much, it will precipitate out of solution and then form an inclusion. There are different examples of different types of inclusions. You do not need to know the different types of inclusions. You just need to know that there are different types of inclusions. Here's some pictures of some inclusions. Magnetosomes shown right there. Metachromatic granules. Let me see if I can blow that up. So there's in yellow the cell, and there's 3 metachromatic granules right here, right there, and right there. This one is sulfur granules inside the cell. This is a sulfur metabolizing cell. So it likes to bring sulfur in the cell and then keep it as a reserve deposit for its needs, its energy needs. And then when it needs energy, it'll metabolize the sulfur. And so this cell, actually all of these cells have a lot of sulfur inside of them, so much so that the sulfur precipitated out of solution, forming the sulfur granules, which are inclusions. Any question about that? So an inclusion can also be called the granule.
So, sorry, just to clarify, when you're saying out of solution, you're meaning that these substances are dense enough that they're not floating in the solution, even though they don't have like an outer membrane or whatever?
Let's talk about the sulfur. Initially, when the sulfur inside the cell is at low concentrations, all of the sulfur will be dissolved in the cytoplasm. There will be no inclusion. But then the cell starts pumping sulfur inside the cell. Eventually, the sulfur will be at high enough concentration inside the cell that it can no longer be in solution. And so the sulfur will precipitate out of solution. If you've ever worked with sugars or salt concentrations in chemistry, you might remember that if you added a certain amount of those molecules to the solution, that the solution could no longer contain it, and then it would precipitate out of solution. Do you remember that?
Okay, yeah, that makes sense.
Okay. Actually, we're at a place to stop, and this is a good place to stop. I don't want to talk about endospores because it'll take at least 5 minutes to talk about that. So let's go ahead and stop the lesson here, and I will continue with this next time. Before I go, are there any questions? All right. I'll see you next time. Remember, there's no
Thank-
lab. You only need to show up if you have questions.
Thank you.
Thank you.
Thank you.
You're welcome.
Thank you.
You're welcome. You know, how do I get that? Not sure. Oh, there we go. Maybe. Having trouble here shutting this down. There we go.